Now we'll go through fat metabolism. So fat metabolism, I probably could have shown a few more things here, but technically fat metabolism is similar in that the first step, similar to carbohydrate metabolism, that the first step is the breakdown of the fat molecule, lipolysis, fat breakdown. And then from there, the fatty acids are going to undergo beta oxidation. The glycerol molecule will actually be converted to pyruvate ultimately, which then makes acetyl-CoA and then passes through Krebs cycle and the electron transport chain. Beta oxidation is also creating acetyl-CoA molecules through a slightly different process. And then those acetyl-CoA molecules will pass through the Krebs cycle, electron transport chain, and oxidative phosphorylation ultimately makes ATP. Two enzymes are involved here, lipoprotein lipase and hormone-sensitive lipase. Lipoprotein, again, this is the substrate on which the enzyme acts, so lipoproteins. So lipoprotein lipase breaks down triglycerides in food and lipoproteins in food. Hormone-sensitive lipase, based on the hormones going around, so maybe glucagon, maybe, that hormone-sensitive lipase would act on triglycerides that are stored in adipose tissue. So again, just another diagram. So adipose tissue, if we were to break down triglycerides through the process of lipolysis, we would release fatty acids and glycerol. Fatty acids would undergo beta oxidation to make acetyl-CoA. Glycerol just makes acetyl-CoA. If it wasn't coming from adipose tissue, say we were going to actually break down some of the food, some of the fat that we ate to use that as energy, then we would still see fatty acids. We'd still see the same thing, fatty acids being converted to acetyl-CoA and glycerol being converted to acetyl-CoA. So remember, lipolysis is just the breakdown of triglyceride into its monomers, fatty acids and glycerol. Fatty acids are metabolized via beta oxidation to make acetyl-CoA. Glycerol is converted to pyruvate to make acetyl-CoA. A couple molecules I want you to be familiar with. Albumin, which is a transport protein. So this is remember that fatty acids are not water soluble, but the blood is water. So in order to get fatty acids from adipose tissue to the cell that needs energy, like a working muscle or the liver cell, we need a way to transport the fatty acid molecule through the blood. So that's albumin. So it is a transport protein, but it's also a phospholipid, so it has a hydrophilic component to it that allows it to move in the blood and carry the fatty acid component on its hydrophobic part. Then once inside the cell, the fatty acid has to get into the mitochondrial membrane to be converted to acetyl-CoA. So carnitine is like a shuttle protein, another transport protein, that's going to move the fatty acid from the cytosol of the cell into the mitochondria. And carnitine is activated by this coenzyme. So the process of beta oxidation, I'll show you a schematic on the next slide. The process of beta oxidation is the process by which we take a full fatty acid chain and basically cleave off 2 carbon segments at a time. So remember, the fatty acid molecule might be, remember we have short chain, medium chain, and long chain fatty acid molecules, and they're just these chains of carbon, 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 carbon bonds. So we might have a fatty acid molecule that's 16 carbons in length, or 18 carbons in length, or 22 carbons in length. Typically, they're always an even number. So beta oxidation is basically cleaving the last 2 carbons from the fatty acid chain. And when we cleave those 2 carbons, we make acetyl-CoA out of those 2 carbons. I didn't show you this diagrammatically, but remember, pyruvate is a 3-carbon molecule. Acetyl-CoA is actually a 2-carbon molecule. So in beta oxidation, we bypass making pyruvate. We directly make acetyl-CoA. So we'll cleave off. So if we have a 16 carbon chain, we're going to cleave off the last 2 carbons, thus leaving us with a 14 carbon chain. And those 2 carbons that we cleaved off are going to be converted into acetyl-CoA, and acetyl-CoA will flow through the Krebs cycle, and then the coenzymes NADH, FADH2 are going to be generated and move into the electron transport chain, etc. So we move from a 16 carbon down to a 14 carbon, and then we're going to take the 14 carbon chain and cleave off another 2 amino acids, leaving us with, or another 2, carbon molecules, leaving us now with a 12 carbon chain. Then we're going to cleave off the last 2 carbons there, leaving us with a 10 carbon chain. Then we're going to cleave off the last 2 carbons, leaving us with an 8 carbon chain, and so on and so on and so on. So if we have a 16 carbon fatty acid chain, we actually can make 18 molecules of acetyl-CoA. And again, if we have a 22 carbon fatty acid chain, we can make 11 molecules of acetyl-CoA. So here is not the greatest, but not a terrible, yeah, it's definitely not the greatest, but it's not a, let's see where it's showing us. So for our class, we don't need to know specifically saturated versus unsaturated fatty acid oxidation. But basically, I want you to see like from the 12 carbon chain down to the 10 carbon chain, right? We basically, it's a little different when we have the double bonds, but just imagine that we just cleaved between carbon 10 and 11. So now we're left with a 10 carbon chain, and these 2 carbons went off to make acetyl-CoA. And ultimately we would cleave all the way down until we had nothing left. Also, if you look at the lecture slides or look in your textbook, there's a much better diagram showing beta oxidation, showing how showing that cleaving of the last 2 carbon molecules. I couldn't find the right diagram to put into this new lecture, but please do refer to the lecture slides to print, because they are from a previous lecture that I wrote, and then refer to your book if you have the textbook to look at some more diagrams of beta oxidation. And then I just want to review why we say that fat is more energy dense than carbohydrate or protein. So remember that 1 gram of fat provides 9 calories, whereas 1 gram of protein or 1 gram of carbohydrate provides only 4 calories. So why is that? Now we know when we have 1 glucose molecule, we only have 6 carbons. That means we can only make 2 acetyl-CoA molecules. If we have even a 12 carbon fatty acid chain, we can make 6 acetyl-CoA molecules. And that's only 1 of 3 fatty acid chains from a triglyceride. And when we say 1 gram of fat, we're talking about 1 gram of triglyceride. So fatty acid chains almost always have more carbon molecules than a glucose or an amino acid, thus leading to more acetyl-CoA production, which ultimately leads to more ATP production. Yeah, as said in other words, there are more carbon-hydrogen bonds compared to glucose, which leads to more ATP production. ATP is our energy source, so if we have more ATP produced per molecule, we have greater energy density. So really anything, any fatty acid chain longer than 6 carbons, so 8 carbon and up, we are producing more acetyl-CoA per molecule. And remember that each triglyceride molecule has 3 fatty acid chains, not just 1. So even if we had 6, even if we only had a 6 carbon chain, if we have 3 of those, then we have 3 times as much acetyl-CoA being produced as 1 glucose molecule. So that's why we say that fat is more energy dense than carbohydrate or protein. And then I want to talk through ketosis and ketoacidosis. So remember, so here's glucose being converted to pyruvate. Here's glycolysis. Pyruvate makes acetyl-CoA, which enters into the Krebs cycle. Remember, pyruvate can also make oxaloacetate. So fatty acids, when they enter, remember they are directly making acetyl-CoA. Glycerol will make pyruvate, but per 1 glycerol molecule, we have 3 fatty acids. So if we only had glycerol and fatty acids, we would wind up with way more acetyl-CoA than oxaloacetate. because the glycerol makes the pyruvate, which makes oxaloacetate. So in the event that we are completely metabolizing triglyceride molecules as an energy substrate, we'll wind up with kind of this backlog of acetyl-CoA. So ultimately, in the process of ketosis, we'll convert acetyl-CoA into what are called ketone bodies. Acetyl-CoA, which might make acetone or might make beta-hydroxybutyrate. So that's basically ketosis, is this process of making these ketone bodies. What's interesting is that we will ultimately take the beta-hydroxybutyrate, one of the ketone bodies, convert it to something called acetoacetate, and back to acetyl-CoA right here, which can then be converted back to acetyl-CoA and enter back into the Krebs cycle. So once we regenerate enough oxaloacetate, we'll actually just convert these ketone bodies back into acetyl-CoA to enter into the Krebs cycle, kind of like the lactate shunting. We just kind of shunt these into these ketone bodies for the time being until we generate enough pyruvate or oxaloacetate to rerun the Krebs cycle. So this is where there's this term that fats burn in a glucose flame or in a carbohydrate flame, and that's because really glucose can also be a substrate for pyruvate or oxaloacetate, just like glycerol. But so in order to really utilize all of the fatty acids that we might have, we really do need to have glucose in order to make enough oxaloacetate. So that's where that term fats burn in a carbohydrate flame comes from. And so this concept of ketosis really is just the accumulation of the ketone bodies in the blood supply. And again, this would happen normally under any sort of feeding conditions if there's high fat content coming into the body. The problem lies if ketosis is going on long enough that it becomes ketoacidosis. So ketoacidosis is where we have enough buildup of these ketone bodies and not enough regeneration of acetyl-CoA, not enough regeneration of oxaloacetate, such that we could convert the ketone bodies to acetyl-CoA to run the Krebs cycle, that we wind up with a buildup of these in the blood supply. These are acids, so they have a very low pH, and so if we have a buildup of them, we might see the blood becoming acidic, and that's what is ketoacidosis. So ketosis, again, the presence of ketone bodies in the blood and the urine. This is happening normally under normal body conditions, typically fairly low amounts of ketone bodies in the blood or urine. These levels of ketone bodies can rise pretty severely. The level of ketone bodies in the blood can rise pretty severely. And that's really where we would see ketoacidosis, is if we have the ketone bodies at very high numbers in the blood or the urine. And this can happen if we see, for example, in the case of type 1 diabetes, if like exogenous insulin isn't provided to the body. We might see the glucose, being converted actually to the acetyl-CoA and then into the ketone bodies, rather than going through the Krebs cycle. So that's where we might see the buildup of the ketone bodies in type 1 diabetes. We might also see it in an event where someone is really trying to just eat exclusively fat molecules and not eating enough glucose substrate or carbohydrate to allow the Krebs cycle to run. So again, ketoacidosis is basically ketosis gone awry, if you will. And so if we do wind up in the state of severe ketosis or ketoacidosis, we'll see dehydration as a symptom because the body's going to try to pee out those ketone bodies. We might also see an increased respiratory rate because we can, in converting some of those ketone bodies, we might be producing a lot of carbon dioxide, so increased respiratory rate to get rid of the CO2. And really, if it goes on for a long period of time, it could be deadly, but typically can be reversed, especially with just a little bit of good complex carbohydrate feeding.